Much like life itself, the nature of science defies simple description. For many years, scientists have written about the scientific method as though there is a single way of doing science. This oversimplification has contributed to confusion on part of non-scientists about the nature of science. So at its core, science is concerned with developing an increasingly accurate understanding of the world around us using observation and reasoning. To begin with, we assume that the natural forces acting now have always acted, that the fundamental nature of the universe has not changed since its inception and is not changing now. So a number of complementary approaches allow understanding of natural phenomena. There is no one scientific method. And this may sound crazy based on what you've been taught and what you've heard, but there's just not one. It's more nuanced than that. Scientists also attempt to be as objective as possible in the interpretation of the data and observations they have collected, because scientists themselves are human. This is not completely possible, but because science is a collective endeavor subject to scrutiny, it itself is self-correcting. One person's results are verified by others, and if the results cannot be repeated, they are rejected. So much of science is descriptive, and the classic vision of the scientific method is that observations lead to hypotheses that in turn make experimentally testable predictions. In this way, we dispassionately evaluate new ideas to arrive at an increasingly accurate view of nature. We discuss this way of doing science later in this very section. But it is important to understand that much of science is purely descriptive. In order to understand anything, the first step is to describe it completely. Much of biology is concerned with arriving at an increasingly accurate description of nature. The study of biodiversity is an example of descriptive science that has implications for other aspects of biology in addition to societal implications. Efforts are currently underway to classify all life on Earth. This ambitious project is purely descriptive, but it will lead to a much greater understanding of biodiversity as well as the effect our species has on biodiversity. One of the most important accomplishments of molecular biology at the dawn of the 21st century was completion of the sequence of the human genome. Many new hypotheses about human biology will be generated by this knowledge, and many experiments will be needed to test these hypotheses, but the determination of the sequence itself was descriptive science. So science uses both deductive and inductive reasoning. The logic recognizes two opposite ways of arriving at logical conclusions, deductive and inductive reasoning. Science makes use of both of these methods, although induction is the primary way of reasoning in hypothesis-driven science. Induction. So deductive reasoning applies general principles to predict specific results. More than 2,200 years ago, the Greek scientist Aristophanes used Euclidean geometry and deductive reasoning to accurately estimate the circumference of the Earth. Please look here now at figure 1.2. Deductive reasoning is the reasoning of mathematics and philosophy. It is used to test the validity of general ideas in all branches of knowledge. For example, if all mammals by definition have hair, you find an animal that does not have hair, then you may conclude that this animal is not a mammal. A biologist uses deductive reasoning to infer the species of a specimen from its characteristics. Inductive reasoning is the logic flows in the opposite direction. I say again, in inductive reasoning, the logic flows in the opposite direction. From specific to general. Inductive reasoning uses specific observations to construct general scientific principles. For example, if poodles have hair and terriers have hair, and every dog that you observe has hair, then you may conclude that all dogs have hair. Inductive reasoning leads to generalizations that can then be tested. Inductive reasoning first became important to science in the 1600s in Europe, when Francis Bacon, Isaac Newton, and others began to use the results of particular experiments to infer general principles about how the world operates. An example from modern biology is the role of specific genes in different studies. Studies on the fruit fly Drosophila melanogaster identified genes that could cause dramatic changes in developmental fate, such as a leg appearing in the place of an antenna. These genes have since been found in essentially all multicellular animals analyzed. This led to the general idea that the same kind of genes control developmental fate in all animals. All thanks a lot to the fruit fly, Drosophila melanogaster. So now let's get to hypothesis-driven science makes and tests predictions. Scientists establish which general principles are true from among the many that might be true through the process of systematically testing alternative proposals. If these proposals have proved inconsistent with experimental observations, then they are rejected as untrue. Take a look at figure 1.3. It illustrates the process. You're seeing it here. After making careful observations, scientists construct a hypothesis, which is a suggested explanation that accounts for those observations. A hypothesis is a proposition that might be true. Those hypotheses that have not yet been disproved are retained. They are useful because they may fit the known facts, but they are always subject to future rejection if, in the light of new information, they are found to be incorrect. This is usually an ongoing process, with a hypothesis changing and being refined with new data. For instance, geneticists George Beadle and Edward Tatum studied the nature of genetic information to arrive at their one gene, one enzyme hypothesis. And we'll get to this later in the course. You'll see this in chapter 15. This hypothesis states that a gene represents the genetic information necessary to make a single enzyme. As investigators learned more about the molecular nature of genetic information, the hypothesis referred to as the one gene, one polypeptide, because enzymes can be made up of more than one polypeptide. With still more information about the nature of genetic information, other investigators found that a single gene can specify more than one polypeptide, and the hypothesis was refined again. So in testing hypotheses, we call that an experiment. So let's get to how we test hypotheses. Suppose you enter a dark room to understand why it is dark. You pose several hypotheses. The first might be, there is no light in the room because the light switch is turned off. An alternative hypothesis might be, there is no light in the room because the light bulb is burned out. And yet another hypothesis might be, I'm going blind. To evaluate these hypotheses, you would conduct an experiment designed to eliminate one or more of the hypotheses. For example, you might test your hypotheses by flipping the light switch. If you do so and the room is still dark, you have disproved the first hypothesis. Something other than the setting of the light switch must be the reason for the darkness. Note that a test such as this does not prove that any of the other hypotheses are true. It merely demonstrates that the one being tested is not. A successful experiment is one in which one or more alternative hypotheses is determined to be inconsistent with the results and thus rejected. So as you proceed through this text, you will encounter many hypotheses that have withstood the test of experiment. Many will continue to do so. Others will be revised as new observations are made by biologists. Biology, like all science, is in a constant state of change, with new ideas appearing and replacing or refining old ones. So keep in mind, a hypothesis must be testable, and it's tested to determine its validity. It is often tested in many different ways, and it allows for predictions to be made. And yes, they can certainly be iterative hypotheses. Establishing controls is next. Often scientists are interested in learning about the processes that are influenced by many factors or variables. To evaluate alternative hypotheses, we keep the variable of interest constant in a control group and alter it in the experimental treatment. This allows us to isolate the effects of a single variable in our experiment, so any differences in the outcome must be from the result of the influence of this variable. Much of the challenge of experimental science lies in designing controls that isolate a particular variable from other factors that might influence a process. Using predictions. For a scientific hypothesis to be successful, it must not only explain and unify observations, but it must also make testable predictions. And that goes right back to what I mentioned moments earlier. When proposing a hypothesis, you need to keep in mind that it may explain everything you have observed, but also be completely wrong. As Thomas Huxley, a contemporary and supporter of Charles Darwin, put it, the great tragedy of science, the slaying of the beautiful theory by an ugly fact. An example from the early history of microbiology involves the observation that nutrient broth left exposed to air becomes contaminated. Two hypotheses were proposed to explain this observation: spontaneous generation and the germ hypothesis. Spontaneous generation held that there was an inherent property in organic molecules that could lead to the spontaneous generation of life. The germ hypothesis that pre-existing organisms that were present in the air could contaminate the nutrient broth. These competing hypotheses were tested by a number of experiments that involved filtering air and boiling broth to kill any contaminating germs. The definitive experiment was performed by Louis Pasteur, who constructed flasks with curved necks that could be exposed to air but would trap any contaminating germs. When such flasks were boiled to sterilize them, they remained sterile. But if the curved neck was broken off, they became contaminated. So this yet again speaks to the importance of hypotheses and using predictions to predict what may and what may not happen upon carrying out some experiment. So as shown here in figure 1.4, you can see how the experiment was carried out by way of Pasteur. This result provides a clear distinction between the two hypotheses. The germ theory predicts the results shown, while spontaneous generation predicts growth in either case. These results then support the germ hypothesis and cause us to reject spontaneous generation. So reductionism breaks larger systems into their component parts. Scientists use the philosophical approach of reductionism to understand the complex system by reducing it to its working parts. Reductionism has been a general approach of biochemistry, which has been enormously successful at unraveling the complexity of cellular metabolism by concentrating on individual pathways and specific enzymes. By analyzing all the pathways and their components, scientists now have an overall picture of the metabolism of cells. Reductionism has limits when applied to living systems. However, one of which is that enzymes do not always behave exactly the same in isolation as they do in their normal cellular context. A larger problem is that the complex interworking of many interconnected functions leads to emergent properties that cannot be predicted based on the workings of their parts. For example, ribosomes are the cellular factories that synthesize proteins, but this function cannot be predicted based on analysis of the individual proteins and the RNA that make up the structure. On a higher level, understanding the physiology of a single Canada goose would not lead to predictions about flocking behavior. The emerging field of systems biology uses mathematical and computational models to deal with the whole as well as understanding the interacting parts. Biologists construct models to explain living systems. So biologists construct models in many different ways for a variety of uses. Geneticists construct models of interacting networks of proteins that control gene expression, often even drawing cartoon figures to represent that which we cannot see. Population biologists build models of how evolutionary change occurs. Cell biologists build models of signal transduction pathways and the events leading from an external sign to internal events. Structural biologists build actual models of the structure of proteins and macromolecular complexes in cells. Models provide a way to organize how we think about a problem. Models can also get us closer to the larger picture and away from the extreme reductionist approach. The working parts are provided by the reductionist analysis, but the model shows how they fit together. Often, these models suggest other experiments that can be performed to refine or test the model. As researchers gain more knowledge about the actual flow of molecules in a living system, more sophisticated kinetic models can be used to apply information about isolated enzymes to their cellular context. In systems biology, this modeling is being applied on a larger scale to regulatory networks during development and even to modeling an entire bacterial cell. So the nature of scientific theories, and this will be ending the chapter. So scientists use the word theory in two main ways. The first meaning of theory is a proposed explanation for some natural phenomenon, often based on some general principle. Thus we speak of the principle first, proposed by Newton, as the theory of gravity. Such theories often bring together concepts that were previously thought to be unrelated. The second meaning of theory is the body of interconnected concepts, and I say again, interconnected concepts, supported by scientific reasoning and experimental evidence that explains the facts in some area of study. Such a theory provides an indispensable framework for organizing a body of knowledge. For example, quantum theory in physics brings together a set of ideas about the nature of the universe. It explains experimental facts and serves as a guide to further questions and experiments. To a scientist, theories are the solid ground of science, expressing ideas of which we are most certain. In contrast to the general public, the word theory usually implies the opposite, a lack of knowledge or a guess. Not surprisingly, this difference often results in confusion. In this text, theory will always be used in a scientific sense in reference to an accepted general principle or body of knowledge. Some critics outside of science attempt to discredit evolution by saying it's just a theory. The hypothesis that evolution has occurred, however, is an accepted scientific fact. It is supported by overwhelming evidence. Modern evolutionary theory is a complex body of ideas, the importance of which spreads far beyond explaining evolution. Its ramifications permeate all areas of biology, and it provides the conceptual framework that unifies biology as a science. Again, the key is how well a hypothesis fits the observations. So evolution theory fits the observations and I mean very well. So research can be basic or applied. In the past, it was fashionable to speak of the scientific method as consisting of an orderly sequence of logical either or steps. Each step would reject one or two mutually incompatible alternatives, as though trial and error testing would inevitably lead the researcher through the maze of uncertainty to the ultimate scientific answer. If this were the case, a computer would make a good scientist. But science is not done this way. As the British philosopher Karl Popper has pointed out, successful scientists, without exception, design their experiments with a pretty fair idea of how the results are going to come out. They have what Popper calls an imaginative preconception of what the truth might be. And again, I say it's an imaginative preconception of what the truth might be. Because insight and imagination play such a large role in the scientific process, some scientists are better at science than others. Just as Kendrick Lamar stands out among songwriters, or the very same way Beyonce may stand out among country songwriters. Some scientists perform basic research, which is intended to extend the boundaries of what we know. These individuals typically work at universities, and their research is usually supported by grants from various agencies and foundations. The information generated by basic research contributes to the growing body of scientific knowledge. It provides the scientific foundation utilized by applied research. Scientists who conduct applied research are often employed by some kind of industry. Their work may involve the manufacture of food additives, the creation of new drugs, or the testing of environmental quality. Research results are published in scientific journals where the experiments and conclusions are reviewed by other scientists. This process of careful evaluation, called peer review, lies at the heart of modern science. It helps to ensure that faculty research or false claims are challenged and not accepted without examination. Recently, there have been problems raised with reproducibility in some areas of biology. That this is being examined indicates the self-reflective nature of science. With some idea of what science is and how it functions, we will consider a single example in detail. What better example than the development of one of the most important ideas in the history of science, Darwin's theory of evolution by means of natural selection. So as we end this section of chapter 1, this is all that has been planned to be included.